Hello, and welcome to Arcadia University's BI327 Histology course. This is the third of the introductory lectures on Introduction to Microscopy. Now, what we've talked about so far is that in order to be able to study the microscopic anatomy, in order to be able to study what's going on at the level of cells and molecules within the human body, we need to have magnification, and we talked about that in the first lecture. We need to be able to process the tissues so we can essentially magnify them. So we essentially use a microscope to be able to increase the apparent size of the image. And we need to be able to go through and add selective contrast so we can look at specific structures that we're interested in studying. The next thing we're going to look at is how the microscope actually works. And so we're going to take a look at the light microscope and then some specialized microscopes that allow us to look at more detail or different aspects of what's going on within a specimen. And so the basic idea of a microscope is that it's going to have some type of illumination source. So in this diagram, we've got a light down there at the bottom in the yellow. It projects the light. We're going to send that through a series of lenses. In this case, we've got a condenser lens, which is in essence going to focus the light illumination source on our specimen. And then we're going to have a series of magnification lenses. We're going to have the objective lens that we talked about previously. We're going to have the eyepiece, the ocular lens, which is going to give us our magnification factor. And so this is what we have in a classic traditional light microscope. We're going to cut our specimen very thin. We're going to put it on a microscope glass slide. We're going to put it on this setup. We're going to shine light through it. And then we're going to put our eye or a camera up there at the top and look at this magnified final image. And this is the basic process of classic microscopy. Now, when we do this, again, it's going to give us a good indication about what's going on within our biological tissue. And if we use some specialized stains, in this case we're using some silver stains combined with hemotoxin and eosin, we can take a look at some skeletal muscle cells, we can see some axons coming down from the nerves, and we can see the motor end plates, where essentially the axon is innervating the muscle, and essentially we're going to have the transmission of the synaptic information. And this is good, but it's limited in what it can tell us. So there are times in which we want to study it and get more information. Now, the second concept, the second microscope we're going to look at is going to be the fluorescent microscope. And we've talked about this very briefly when we talked about the antibody staining and using fluorescent molecules. So in this case, instead of using a visible light source, we're going to use a fluorescent wavelength of light. Still visible, but it's a much narrower beam, a light that's coming through, narrow range. And it has a different, essentially, wavelength associated with it. We're still going to be using glass lenses, and it has the same basic setup as our traditional light microscope. But this, in essence, allows us to see fluorescent molecules. So either autofluorescence, there's certain structures within the body that if you illuminate them with a fluorescent light wavelength, they're going to glow on their own. Or, as we talked in the immunohistochemistry, immunocytochemistry, the antibody staining, we can go through and see where a specific protein is going to be located. But the same basic idea of what we're doing with a light microscope. And again, using our example here, we can use our fluorescent microscope and with antibodies see acetylcholine receptors, essentially the muscle protein that receives that synaptic information, glowing in red, and the nerve terminal, the axon in essence, the tip of the axon, is going to be glowing in green because of the properties there. So we can look at this and see a little bit more detail about what's going on. And if we're interested in acetylcholine receptors, we can study very specifically what those receptors are doing on that muscle cell membrane. Now, that was our traditional fluorescent microscope. There's also a confocal microscope. And the confocal microscope, in essence, advances what we can do with a fluorescent microscope by essentially using a very, very tight, almost like a laser beam, and it is a laser beam, point of illumination. We're still using the glass lenses, but instead of a broad illumination that's projecting on the entire specimen, we're essentially projecting like a laser onto a very, very narrow portion of our specimen. And it's essentially going to allow us to focus our light beam our fluorescent beam onto a specific region, a specific very narrow region of our specimen. And it's going to be described as optical sectioning because we're essentially only illuminating a very tight region of our specimen. And so when we take a look at this, what we can see then is a much clearer image because we don't have other things either above or below the plane that we're looking at. The amount of tissue that we're looking at isn't obscuring, isn't blurring the image, and we've got a very, very fine focus point. And so in this case, again, looking at the same basic concept, neuromuscular junction are red, synaptic vesicles in green, and our nerves in blue. And by using the confocal microscope hooked up to a computer, we can in essence come up with three-dimensional images by essentially taking very narrow slices through our biological specimen and then reconstructing them using the computer. And so this is an advancement on what we had with fluorescent microscopy. Other advancements in microscopy, we said we wanted to be able to look at a much higher resolution. And so one way to do that would be to use a narrower, essentially a shorter wavelength of light. And so one way to do that is instead of using light waves or even fluorescent light waves, is to use an electron beam with a very, very short wavelength. Now, the electron beam can be generated, but our glass lenses would absorb all of the energy of that. And so instead of a glass lens, we're going to use electromagnets to focus our electron beam. And instead of colored chromogens, either fluorescent molecules or chemical molecules, we're going to be looking at adding our contrast with heavy metal stains. Things like uranyl acetate or lead citrate are going to be heavy metals that are going to bind to specific structures within biological organisms, biological tissues, and they'll essentially absorb our electron beam so that what we're going to be looking at are going to be shadows that are going to be projected up into where we're going to be visualizing it, whether it's a screen that we're going to be looking at or in essence a camera that's going to be collecting the images. As we said, using the transmission electron microscopy, transmission because it's projecting the electron beam through the biological tissue, we're going to have much thinner sections, we're going to have a much shorter wavelength of light, so we're going to have a much higher resolution associated with it. We can see things at about 1,000 times greater detail than what we can see with a light microscope. And so you can go through, and again, in this case, we're looking at a neuromuscular junction. You can see the muscle membrane on the bottom portion of it. You can see the nerve terminal marked with T at the top portion of it, and you actually see the membrane components. You can see the synaptic vesicles within the nerve terminal that has the acetylcholine that's going to be released to trigger the muscle cell to contract. You can see that at a level that's not possible with the light microscope. Okay, so this is transmission electron microscope shooting our electron beam through our biological specimen and essentially looking at a shadow that's created from the staining pattern. We can also do scanning electron microscopy, and instead of projecting our electron beam through our biological specimen, we're essentially going to bounce the light, in this case bounce the electron beam, our illumination source, off of the object. And this is going to allow us to take a look at surfaces that are going to be present. And so in this case, instead of slicing the tissue, we're going to look at a preserved portion of it. We're going to coat the external surface with a metal to scatter the electrons that are being shot at it. And again, taking a look at our neuromuscular junction, this can allow us then to get a 3D, three-dimensional visualization of the surfaces of the tissue. And so in this case, we can see the relatively large cylindrical muscle fibers. We can see the nerve coming down almost like a rope that's kind of untwisted and then interacting at the motor end plate to show us how the nerve is going to be coming into contact with the muscle. And again, gives us some more information that we can use to better understand what's going on within our biological specimens. But with the set of slides, we've looked at various ways in which we can look at and learn about the connection between a nerve and a muscle cell and better understand what is going on, better understand what the proteins are doing, better understand what that mechanism is, that relationship between anatomy and physiology by using these different tools. That's the end of the Introduction to Microscopy lecture. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks.